Hi, I'm Jan Witkowski. I'm Director of the Banbury Centre here at Cold Spring Harbour Laboratory. Welcome on this rather brightening but definitely gloomy afternoon here at Cold Spring Harbour. This is the 81st annual symposium, and the topic this year is targeting cancer. And I have Bill Kaelin here, who's at Dana-Farber and also a Howard Hughes investigator. Targeting cancer, Bill, how does that relate to your work, or how does your work to relate to the topic of the symposium?
Yes. Well, as a postdoctoral fellow, I had worked on the retinoblastoma tumor suppressor gene, and I think it was through that experience that I really learned the power of letting the cancer genetics help guide you in terms of what pathways and molecules they hold near and dear. And so when I started my own laboratory in the early 1990s, I came across a paper on the identification of the von Hippel-Lindau, or VHL, tumor suppressor gene. And my work as a clinician when I was younger had taught me that the cancers that von Hippel-Lindau gene mutation carriers develop, such as kidney cancers and hemangioblastomas, are very rich in blood vessels, and they also sometimes cause the patient to produce an excess number of red blood cells. And when you think about it, what those two events have in common is that those would normally be the responses if a tissue was starved of oxygen. You try to bring in more blood vessels and you try to bring in more red blood cells. And so we thought by studying the von Hippel-Lindau gene, we'd learn something about oxygen sensing, and we thought we'd also learn something about the pathogenesis of kidney cancer and other cancers caused by VHL mutations. And that's been a road I've been on for about 20 some odd years now, but the good news is I think in the course of it we learned something about how the body does indeed sense changes in oxygen availability through a transcription factor called HIF, and modifications of HIF that take place in an oxygen-dependent manner, and how that governs whether the VHL protein does or does not destroy the HIF protein. And that's led to some interesting and new ways of approaching diseases characterized by inadequate oxygen delivery. But for our topic of discussion today, it also taught us that kidney cancers care very deeply about one particular HIF, namely HIF2. And HIF2 in turn turns on various genes, including the vascular endothelial growth factor. And I think that knowledge helped to sort of accelerate the development of VEGF inhibitors for kidney cancer, which have now been FDA approved. And we've also been working with a biotech company called Peloton that's done a marvelous job developing the first generation of small molecules that directly target HIF2 itself, so the master regulator. And these have just entered clinical trials, and I think it's very early days, but it looks like there may be some activity there as well. And so as I started out mentioning, I'm a big believer in genetically validated pathways as being the very fertile area for looking for drug targets. Our work on VHL and oxygen sensing and more broadly metabolism led us to an interest in the cancers that are caused by mutations in isocitrate dehydrogenase 1 or 2. And we learned over the years some work done at a company called Agios that these mutant proteins actually overproduce a abnormal metabolite, a so-called oncometabolite, which we call 2HG for short. And we and many other labs around the world have begun to understand how the 2HG causes cancer, and this has also helped to provide a framework for the first generation of drugs that now prevent the production of 2-hydroxyglutarate, and at least in leukemias caused by IDH mutations, this is again leading to some very dramatic clinical responses.
You're mentioning blood vessels, of course, makes me think of Judah Folkman
Yes.
and his angiogenesis. How do Judah's ideas fit with yours? Because as I recall, Judah thought that you're saying that the increase in blood vessels is a consequence of lowered oxygen, whereas I seem to recall that Judah rather thought that maybe I'm completely wrong, that the proliferation of blood vessels was in a way the cause, or
I think it's an excellent question. So I think actually even long before Judah Folkman, some of the great German pathologists in the 19th century appreciated that all solid tumors have regions that are hypoxic, and hypothesized, as did Judah, that if we could simply block these new blood vessels from supplying blood to these tumors, it would be a useful way to control their growth. So I think the general idea that blocking angiogenesis would have anti-cancer activity is correct. What we had going for us in the kidney cancer setting is you had a genetic smoking gun, that here's a tumor that you can almost be sure is causing its angiogenesis by upregulating HIF and in particular VEGF. And the reason that's important is that there are actually a host of factors that can induce angiogenesis, and in many tumors there's probably some chicken soup combination, whereas in kidney cancer you can be quite sure that VEGF is going to be doing most of the work. And so I think that's why if you look at solid tumors, there are other solid tumors that will respond to VEGF inhibitors, but the responses are much more significant in kidney cancer. I will also say the other thing that this story illustrates is, although Judah was certainly correct in terms of saying we need to develop angiogenesis inhibitors, the inhibitors he was developing were never really grounded in a firm molecular and genetic understanding of which growth factors are most essential for inducing blood vessels. That came later with the work of Napoleone Ferrara and others who taught us through mouse engineering and other studies that VEGF is clearly one of the most significant factors used by tumors to make blood vessels.
So why? If you block VEGF in a kidney tumor,
Yes.
can't it use these other pathways to? Are there other pathways that it could use to increase angiogenesis?
Yeah, I think that's a very important question. So, although VEGF inhibitors are very active in kidney cancer, and some patients will go into durable remissions that can be measured in years, to my knowledge, no one has been cured with a VEGF inhibitor. They do inevitably relapse, and it's quite likely that, as you're hinting, that this is due to workarounds or collateral pathways being activated that provide other means of inducing angiogenesis. And so if we understood more about what those factors might be, then we could obviously perhaps build better combinations.
So let me ask you a question. We can use it as a transition to the other things that we were talking about. So you began work in this area 20...
I think I started around 1993, so
23 years.
Yeah.
And that, of course, it begs the question about the length of time it takes from some fundamental discovery of mechanisms of the cell to having some molecule that the FDA approves and goes into. And I know you're very concerned that one of the factors that may be involved in this is that the academic research is not, and we can discuss precisely what the word is, reproducible, or certainly it's not of a level that a pharma thinks that they can just take at face value.
Yes. Well, I think there are several layers to that question. So one thing that has impressed me over time, watching the drug discovery and drug development process from at least the sidelines, since I do work in academia and not in pharma, is there's a big difference between targets and strategies where, again, you have a genetic smoking gun, preferably in people, but if not in people, at least in a relevant animal model such as the mouse, that this should be a target, as opposed to things that are based on more circumstantial evidence, but where you don't have that genetic smoking gun. So as I said, in terms of developing angiogenesis inhibitors, VEGF was the first angiogenic strategy that in my mind was really based on a molecular understanding of angiogenesis, and in particular where you had that genetic smoking gun. I think the process of going from target to an actual drug is accelerating when you do have a high value target where you maybe have genetic evidence, but also hopefully have other corroborating lines of evidence that this should be a target, and where the basic findings have proven to be robust, meaning other investigators in the field, either in academia or pharma or both, have been able to replicate, the essence of the published work. And what you're hinting at, and what I talked a little bit about last night, is unfortunately our colleagues in pharma and biotech are telling us that more frequently than we would like, they find that either the published work related to targets emerging from academic labs is either not reproducible, or more commonly reproducible but not robust, meaning it's true in a set of defined circumstances, but those circumstances are sufficiently narrow that it's unlikely to translate into a meaningful new drug. And so I think we have to have a greater appreciation that for something to really lead to a drug discovery effort, we need to emphasize robustness.
So I think this issue of replication and really the difference between academic research, and I think there is a difference between academic research and pharma research. Presumably, I shouldn't say presumably, in academic research I can imagine that a piece of work is done and conclusions are drawn. That work may not be replicated, as pH by pH. Or,
Yes.
but there are other findings that are based or related to that finding that back it up.
Yes.
Now, is that sufficient? Is that sufficient foundation for? Is that a sort of replication,
Yes.
sort of reproduction of that original finding?
Yes, I mean, I think
Corollary evidence,
I think
maybe.
I think that the key is, as you say, are there corroborating lines of evidence to support the conclusion, and has that conclusion made predictions that perhaps you otherwise would not have made that then turn out to be true? Of course,
Yes.
that's at the heart of the scientific method and at the heart of scientific progress. I think unfortunately the publication event has become sort of the golden ring in this process, and we have these very talented and ambitious trainees, and they see that as the end to the means, getting that publication.
Absolutely.
I think we have to step back and remember that, as you say, it's really about, will the work stand the test of time? Will it enable other people to build upon it? And I think that's what we have to do a better job emphasizing to our young people.
But more generally, the goals of academic research, well, I suppose you've just covered it in a way, that the trainees need to get publications, but academic research and research in pharma do have different goals.
Yes.
And somehow to require, or for pharma to expect the academic research to, I don't want to use pejorative
Yes.
words, to be like pharma
Yeah.
research is, I would have thought, really is not going to be met.
Yes. Well, I think you bring up again another really important point. I think ideally we would form a relationship with pharma and biotech that would be symbiotic and complementary and would build upon each other's strengths. And as you point out correctly, I think there are things that, frankly, we perhaps do better than they do, but at the same time, there are things that I would acknowledge they do better than we do. So I think we have to understand that sort of sociology and that sort of handoff. I also think one thing that's happened over the past 10 or 20 years is, I think, to me, the secret sauce of American biomedicine was that appreciation and understanding that we would rely on the academic investigators to do classical, curiosity-driven research without putting a gun to their head saying, we want to see the application, we want to see the deliverables. You do your curiosity-driven research, learn the fundamental principles of the system, and if on occasion that leads to sort of a translatable moment where you have something you could build upon, maybe to do the handoff to our colleagues in biotech and pharma. I think, unfortunately, implicitly or explicitly, we've been coercing academic scientists to become more translational, to become more applied. So I think one of the problems we've got into is, frankly, we now have people, in some cases, doing science they probably shouldn't be doing. It's not what they would naturally be more inclined to do.
Or even given the pressure you've just said about that institutions and universities require or expect, and funding bodies expect it too, that perhaps people make claims before, and you mentioned this earlier, they make claims about their research, not about whether it's right or not, but claims about its applicability.
Categorically true. I mean, I think this is really, now we're getting close to the heart of the problem. I mentioned last night that when I read papers now in figures 1 or 2, maybe there's some interesting phenomenon that in years past would have been more fully developed from a mechanistic and fundamental point of view, but you can almost see the race begin to get to that last figure, which is going to be some gratuitous attempt at clinical relevance, and it's going to be a contrived, therapeutic preclinical experiment, or maybe some correlative data on patient outcomes, or some attempt to justify the work. And it sometimes makes me want to cry because, again, I think, as you say, it's people trying to justify what they're doing, and I think it's actually having some unintended negative consequences.
So in the last couple of minutes, do you see a way to deal with this issue?
Well, I think we as a scientific community have to really have a dialogue about this relationship we've discussed, what we in academia should or should not be attempting to do, what standards we should hold one another to, particularly when it comes to what constitutes a publishable set of observations. And I've said only half jokingly that a paper 20 years ago was one thing proven 6 ways, and I think slowly we've moved towards papers being 6 things proven 1 way, and I think it becomes a house of cards, and it leads to this robustness problem. So I think we need to have a serious conversation amongst ourselves, because at the end of the day, we're reviewing each other's papers. We do, I think, have a lot of influence in terms of what gets published and what constitutes a publishable event. I had a conversation along these lines just an hour ago with an editor, and I think it's always sort of easy to reflexively say, oh, I'd like to see one more experiment, but I think it does become a bridge too far. Frankly, I think the NIH director and the NCI director have to, and other people in Bethesda and Washington also have to use their bully pulpits to say enough is enough. This is what we now expect of the scientific community, given these resources that we've been entrusted with.
Well, that seems to be the right moment to end.
Okay.
Thanks very much, Bill.
Thank you.
Thank you.
Okay, thanks. All righty.